On this slide, you will see listed the objectives for our innate immunity module. Today is the third of 5 mini lectures that will cover this material, and today's lecture will focus on the local inflammatory response. So here I've shown a picture of an epithelial barrier that has been compromised and some bacteria that are now infecting the underlying tissue, as well as a local capillary. And as we've mentioned before, there are macrophages within that tissue that express pattern recognition receptors that can recognize pathogen-associated molecular patterns expressed by these bacteria. And when that happens, these macrophages start to produce cytokines such as IL-1, TNF, and IL-6, as well as chemokines such as IL-8. And remember that at the same time, these bacteria are being exposed to complement within the interstitial fluid, and part of the result of that is the production of C3a and C5a. These complement components and these cytokines act upon these endothelial cells to induce their activation. These endothelial cells express both complement receptors and cytokine receptors. One of the things that happens when these molecules act and activate endothelial cells is these endothelial cells start to produce their own chemokines within the capillary. And one of the main chemokines that they produce is interleukin 8. The other thing that these molecules induce is the expression of certain adhesion molecules called selectins and ICAMs. And what happens when these are expressed is, also let me mention that these cytokines, especially IL-1 and TNF, induce vasodilation of the capillary. And so this enhances the ability of cells to exit from circulation into the inflamed tissue. So one of the things that happens when these endothelial cells become activated and start to express these integrins and these ICAMs is it slows down the rolling of neutrophils along the capillary bed. The endothelial cells become sticky. As that happens, this allows those neutrophils to start to bind to the chemokines being produced by these endothelial cells via their chemokine receptors. When that happens, this induces the expression of a high-affinity integrin called LFA1. When LFA1 is expressed, it causes these neutrophils, or really any cell type that's circulating, to bind firmly to these endothelial cells and adhere. And so here we have neutrophils that are now smushed up against this endothelial cell wall and are firmly bound. And these selectins, integrins, ICAMs, and adressins, they really control migration across these endothelial barriers. These are molecules that are adhesion molecules that mediate stickiness. Again, as I mentioned, the selectins and the ICAMs help slow down the roll of the neutrophils along the capillary bed and allow for those neutrophils to bind to the chemokine interleukin 8 to express the high affinity version of LFA1 and firmly adhere. And as you can see, there's a lot of family members, and I'm not really going to hold you responsible for all of these family members. I do want you to remember LFA1 as a key integrin that mediates this process. Once these cells firmly adhere, they can begin the process of diapedesis, in which they transmigrate in between the tight junctions and into the tissue. This is a video that I'm going to show that if you look closely, you'll see an arrow pointing to a rolling white blood cell that is going to now firmly adhere right here. And you can see it kind of disappear and kind of smush up against the cell wall. It is now migrating across this endothelial barrier, and you're about to see it pop out right here on the other side of that endothelial cell wall. So this is a process called diapedesis. Oops. So neutrophils, once they have migrated into the tissue, now can migrate along a chemokine gradient to the site of inflammation. So here we have a couple of macrophages that are making interleukin 8. The concentration of interleukin 8 within that tissue is highest the closer you are to those macrophages. And the farther away you get, the lower that concentration. Neutrophils can bind to IL-8 via chemokine receptors, and this induces their migration along this chemokine gradient until they reach that site of inflammation. This is a good example of how chemokines work. This is, or chemotaxis in general works. This is a end of a pin or a needle that's been coated with a bacterial peptide called N-formylated methionine. Neutrophils have receptors for this bacterial peptide that cause them to migrate towards it. And so you'll see that these neutrophils will start to migrate towards the source of this FMLP, and it's actually a pretty rapid and impressive process. And so this is the process of chemotaxis. There are many chemokines, and chemokines have a variety of important functions. They mediate migration to inflamed tissues. They mediate trafficking of immune cells within secondary lymphoid organs and organization of those organs. They mediate interactions of T cells with antigen-presenting cells, migration of T cells into B cell follicles and germinal centers. So basically, without chemokines, you don't get an inflammatory response, you don't get an adaptive immune response, and you don't have lymphoid organs. What you'll notice from this chart, and I obviously don't want you to memorize this chart, but what I do want you to take away is that different chemokines are associated with the recruitment of different cell types. So some of these chemokines recruit neutrophils. So here's a bunch of chemokines that recruit neutrophils, including IL-8. Other of these chemokines recruit T cells, B cells, eosinophils, and so the flavor of the inflammatory response, the chemokines that are induced during the inflammatory response influence the nature of the cells that migrate to that site of inflammation. So here's a summary of the early inflammatory response. The hallmarks of inflammation are redness, swelling, heat, and pain, and they can really be explained by the increased migration of blood and white blood cells into sites of inflammation. Macrophages reside in tissues. They don't circulate. They are the first line of defense. They initiate the inflammatory response. Mast cells are also another cell type that are tissue resident and can contribute to this. What we call the first responders are typically the neutrophils, and these are cells that are the first to migrate from circulation into the inflamed tissue. So the macrophages secrete cytokines, put up their warning signals, and induce the migration of neutrophils into that tissue. Also, some monocytes will migrate into those inflamed tissues. Monocytes, however, once they migrate into that tissue, differentiate into tissue macrophages within that site of inflammation. Other white blood cells use very similar mechanisms to migrate into those sites of inflammation, but the cellular makeup of the inflammatory infiltrate is dependent on the chemokines that are present. Okay, so here are some of the key points from this mini lecture. You'll want to go review the material and make sure that you have captured all the information that you need.